Well, good morning, everyone, and welcome to today's webcast. My name is Josh Forsyth. I am the Vice President of Marketing here at Golden Helix. You know, we've been doing these webcasts for many years now. Never ceases to amaze me how many people show up for them, so it's been very encouraging. For today's webcast, we had nearly 400 register, so just wanted to thank everyone that has attended now and in years past for their support. I'm grateful today to be joined by one of our customers, Mr. Khanh-Nhat Tran-Viet of the Duke University Medical Center. Mr. Tran-Viet is the manager and a research analyst in Professor Terry Young's lab there, which has been globally involved in covering a variety of association and gene discovery studies for ophthalmic disease. Today, Mr. Tran-Viet will be discussing various aspects of the lab's research into rare ocular diseases and how they have been able to utilize exome sequencing technologies and Golden Helix software to find potentially pathogenic variants. My hope is that by the end of the webinar, you will have a good sense of not only how our tools can be applied to next generation sequencing, but also have a better understanding of the technology and methods involved. Before we begin, a couple of logistical issues. First, we have set aside an hour for this webcast. Our plan is to present for about 45 minutes or so, and then we will have a question and answer session at the end. We encourage you to ask questions throughout the webcast, and at the end, to do so, there is a question and answer pane that comes with the GoToMeeting software here. So please type in your questions there, and we will get them to them during the Q&A portion of the webcast. With that, I will turn the presentation over to Mr. Tran-Viet.
Thank you, Josh, for the introduction, and it's an honor to share some of our experiences with current and potential future users of Golden Helix. Thanks to all of you who've joined today, and hopefully the webcast will be informative and will provide you with some of the insights on to how to apply some of these tools offered in SVS in your own studies and research. We purchased this software about 6 months ago, and we've been fairly pleased with it thus far. And I think like any other software tool, there's a little bit of learning curve, but I think myself and the rest of the staff have found it pretty useful than what we actually used to do, which was kind of using Excel to filter candidate variants. So this is sort of the agenda for today that I'd like to cover. First, I'll briefly go over the landscape of NGS, some background on who we are and what we do, and then talk about our experiences using Golden Helix SVS software by showing you tools we were able to implement and utilize in one of our studies to find that needle in the haystack, if you will. I'd also like to preface by saying that if you haven't watched Dr. Kaufman's talk on how to filter variants from last summer, I'd highly encourage it you do so, as he also touches on some of the basic tools out there to help filter variants of interest. Here I'll use some of the newest public databases, I think which will be released this afternoon on the SVS 7.7.4. To filter in an actual case, and I'll specifically go over how to use tools to filter using marker statistics, gene lists, functional predictions, and multiple columns. And lastly, I'll cover what I think are some opportunities and challenges that we all face and conclude with any questions that you may have. So whole exome sequencing is a robust technology that's extremely practical for investigating coding variations on a genome-wide level. I think with countless service providers out there now that can help you run the experiment, store it, and analyze it, it's created a huge push for not only the scientific community, but also from the healthcare sector to interpret data such as exome sequencing, which can perhaps be applied to reach the end goal of understanding the genetic etiology of human diseases. Certainly the ability of large number of patient samples to be screened in a robust fashion is the attraction I think that NGS brings. And even in discovery, especially in human genetics research. With more and more media exposure of understanding your genome, I think there's been quite a bit of news in the past several years in obtaining one's blueprint, and even inquisitive celebrities such as Larry King, Ozzy Osbourne, and Glenn Close has had their genome mapped, actually. And the figure you see there in the top right corner actually gives you a glimpse of the increasing number of whole exome sequencing papers that's come out since 2010. And it's obviously continuing to grow as more and more diseases are discovered, and as we get a better grasp of not only understanding the technological advancements, but also the pipelines involved in accurately detecting mutations. So a brief background on who we are, and as Josh sort of mentioned earlier, we're part of the Center for Human Genetics at Duke University Medical Center, and our group's led by Dr. Terry Young. She's a professor of pediatric ophthalmology and medicine and has faculty appointments not only at the center, but also at the Duke Eye Center and at Duke National University of Singapore Graduate Medical School. Our interest in ophthalmic genetics focuses on complex diseases as well as rare Mendelian diseases. I think over the past several years, there's been significant advances in understanding the complexities of ocular genetics, and our recent involvement in worldwide collaborative studies have hopefully shed some light into this field. This is certainly not an exhaustive list, but some of our current studies we are actively involved in includes myopia, primary congenital glaucoma, Stickler-Wagner syndromes, corneal dystrophy, strabismus, eyelid malformation, and microphthalmia and ophthalmia, where children are actually born with small or no eyes at all. And some of these are, I think Napoleon Dynamite and Harry Potter all wear glasses, so they would be part of the study. And Steve Nash from the Los Angeles Lakers, in fact, is cross-eyed. But why expend resources on vision research, right? Visual impairment is often regarded as a benign disorder due to efficient treatment options, such as glasses, contact lenses, and surgery. And it's not always recognized as a disease phenotype in medical registries. Well, with multiple aggregated studies out there now, we now know that vision impairment is a huge economic burden in the United States. There was a study done in 2007 where it's estimated that the annual cost of vision problems in the US is actually approximately $51.4 billion. Though a huge chunk of the costs are accounted for by direct medical costs, we see that health utility, which measures the quality of life for those who are affected and the surrounding loved one, actually accounts for a good portion of this cost, highlighted in blue there at $10.5 billion annually. On average, we adults in the United States spend approximately $200 annually on myopia-related costs, such as doctor visits, glasses, and contacts, and which as a nation exceeds $14 billion annually. So who's affected, right? If you or the people around you wear glasses or to correct your vision or even wear contacts, unfortunately, I hate to say it, but you're part of the statistics. As a reference point, and worth pointing out that more than 12 million school-aged children has some sort of vision impairment, and 1 in 20 can have vision problems that can lead to permanent sight loss if untreated. Some of these eye problems seen in children are myopia, strabismus, and amblyopia. I also want to point out here that vision impairment is not just affecting people in the United States, but also around the world. Here's a table that summarizes literature comparing myopia prevalence rates. About 33% of the people in the US have some degree of myopia, but you can see that studies in both Taiwan and Hong Kong have shown that there's greater than 80% of the population are perhaps myopic, and vision impairment is actually considered an epidemic in some of these countries. Well, so with twin studies and animal experiments using tree shrew and chicks, we can pair this up with epidemiological studies out there now, which then provide us clues with some ocular conditions such as myopia, and tells us that it's not only genetic, but environmental as well. Today, the exome data we'll actually walk through is a disease called Stickler Syndrome, which presents itself with an array of hereditary conditions. Stickler Syndrome is a group of conditions characterized by ocular abnormalities, hearing loss, joint problems, and distinctive facial appearance. These signs and symptoms vary among affected individuals in a family, and oftentimes can be underdiagnosed and misdiagnosed by different specialists. And estimates actually show that Stickler Syndrome affects 1 in 10,000 newborns. The reason why we're actually interested in this syndrome obviously is for the ocular manifestations that are sometimes seen with these patients. Typical ocular presentations within Stickler Syndrome include severe nearsightedness, or what we call high-grade myopia, and other eye problems such as cataract and retinal detachment, and even vitreous abnormalities, which are kind of like the clear gel that fills the eyeballs, can also be affected. To date, 5 genes have actually been identified for Stickler Syndrome. Mutations in COL2A1, 11A1, 11A2, 9A1, and 9A2 cause Stickler Syndromes types 1 through 5, where all the genes listed above are involved in the production of 3 types of collagen, type 2, 9, and 11. Collagens are complex molecules that provide structure and strength to connective tissues that support the body's joints and organs. Type 2, 9, and 11 collagens are components of the vitreous, cartilage, and other connective tissues. Thus, biologically, we know that it makes sense that mutations in any of these genes impair the production, processing, or assembly of type 2, 9, and 11 collagen. Defective collagen molecules or reduced amounts of collagen disrupt the development of connective tissues, leading to characteristic features of Stickler Syndrome. Though these genes account for the majority of Stickler syndrome patients, there have been reported cases where no mutations were discovered in any of these known genes. Thus, there is some thought that mutations in other genes may also cause this condition, but those genes haven't been identified yet. So we were actually initially referred to by an ophthalmologist in North Dakota who clinically diagnosed several individuals from this family as Wagner Syndrome, where ocular manifestations were very similar to Stickler, and the initial patients he was caring for had no other systemic manifestations. The family had molecular testing done on no mutations on all the collagen genes, and no mutations were actually discovered. But as we recruited more family members across multiple states, we realized that clinical presentations varied among these individuals, where some individuals had more skeletal issues such as scoliosis, requiring braces instead of myopia, or other ocular conditions. We ended up collecting about 49 family members with 16 affecteds, and we chose 2 samples for exome sequencing. And this here kind of depicts the processes that we went through. We ended up sending 2 samples off for exome sequencing using NimbleGem V2 library capture kit with an approximately 50x mean depth coverage and using Hudson Alpha's services. The data was aligned, duplicates were removed, and GATK was used to call the variants. We have a bioinformatics team in Singapore that helps with this portion, but we also enlisted the use of Hudson Alpha's bioinformatics team to generate the data as well. One of the types of input data required for using SVS is a VCF file. So if you don't have experience in running scripts for the bioinformatics portion, I'd highly encourage you to use some of the services out there that can be included as part of your quote. All said and done, I think our output file we'll need for analysis consists of the VCF, which is a variant calling file, and then the BAM files, which you also need to, if you decide to visualize the sequences. And you can use Golden Helix's freeware called Genome Browse for that. And the 2 samples that you see highlighted on the very bottom left and on the right are the 2 individuals that we chose for exome sequencing. And so once we obtained the files we needed for filtering, a couple of assumptions that we had to make or we wanted to make to get our minds right for filtering. I think we're going to try to go after a rare variant, which based on the affected status of individuals in this family showing an autosomal dominant model, and hoping that the variant would actually alter the amino acid, and which is also conserved across multiple species and is also biologically relevant to the disease. And so this is kind of a pipeline overview of what exactly we did with filtering using SVS. And if we zoom in here, we took 6 major steps in getting the variants down from 73,000 to 9. We started with 73,000 variants across 2 affected individuals. And for the first 3 steps, I'm not going to go into detail about explaining in detail, just simply because I think these are tools that's also included in the protocols that Golden Helix has on their website. But essentially, the first thing we actually wanted to do was we only wanted to look at the regions that had exons and removed introns and intergenic variants that were identified. So we actually kept everything that's plus or minus 10 just in case we wanted to keep the splice sites. And we had the variants down to 25,485 at that point. And then next, what we wanted to do was we're still looking for rare variants here. So we wanted to use common DV-SNP 137 database, which excludes everything that's greater than 1% of the population in terms of allele frequencies. And so once we kicked that in, we ended up with 3,071 variants. And then we did a variant classification tool that Golden Helix has, which classifies variants as either coding, intronic, synonymous, et cetera, et cetera. And we actually chose all the variants except for synonymous. So we kept anything that's splice sites or any of that sort. And once we did that, we got it down to 1,900 variants. So from this point on, the 3 tools that I felt were really useful that perhaps you guys can use in your own analysis is filter by marker statistics, filter by gene list, and also filter by functional prediction sites. And so the first thing we'll kind of go over is filter by marker statistics. Marker statistics is a very useful tool, we felt like, because it actually uses some of the genotype information that are provided in your VCF file. And you can actually delineate or differentiate samples or groups of samples based on the call rates or even frequencies of genotypes of interest. And so I kind of wrote it in red there so that you guys can actually kind of see it. But basically call rates, what we kind of want to answer the question is an X number of my samples have to have some sort of a genotype call. frequencies of genotypes of interest actually kind of says, hey, X number of samples have or don't have to have a certain type of genotype calls. So in our case, if we're looking for something rare and we're looking for something that's inherited in an autosomal dominant fashion, that we want to make sure that both variants are being called, and then also the frequencies of the genotype that we're actually chasing after should be a heterozygous mutation. And here's an outline of the tools here that you can actually use and click and go over. But let me walk you guys through the steps and see what's going on. So I've already done the first 3 filtering criteria up here. And so again, I'm not going to highlight that. However, the starting material that we do have is 2 samples, affected 1 and 2, at 1,900 variants. So if we want to do marker statistics, you want to go under quality assurance, genotype statistics by marker. And it's going to ask you if it's not going to contain non-autosomal markers, just say yes. And then you're going to make sure that you classify alleles by reference and alternates. And you can check any number of these information, but I'm just going to exclude out Hardy, Weinberg, Fisher exact tests just for the purpose of this webcast. And so once we hit run, we should get a list of variants with all the information that the marker statistics tools does. And it gives you call rates, negative log 10 call rates, number of distinct alleles, alternate allele frequencies, and then genotype counts, et cetera, et cetera, et cetera. So the 2 things that we really want to pull out of this marker statistics is, 1, we want to say that both individuals have to have a call at some point in time, a call at that position. And the reason why that's important is because, perhaps there could have been one sample may not have the variant call just simply due to the fact that perhaps the depth was too low to even make a call or something like that. So again, this filtering kind of goes through the most stringent probably criteria, but just as a kind of walkthrough and see how you guys can manipulate it later down the line, hopefully you'll see some usage here. So for call rates, it's delineating between, obviously there's only 2 samples, so it's either 0% where no calls are being made, or 0.5, which is 50% of the samples are being called, or 1. And in our case, we're looking for something that's 100%. So you can actually right-click on any of these columns, and in this case, I'm going to highlight, right-click the call rate, and you can do a function called activate by threshold. And activate by threshold, again, kind of says, okay, well, I want it less than, greater than, equal to, et cetera, et cetera, et cetera, to the value that you want. In our case, we know exactly that we want 100% call rate. So we're going to type in equals to 1. And just keep a note up here on the top right. We're starting with 1,900 variants. And if we say we only want to look at the variants that have 100% calls, hit run, and that 1,900 variants actually drops down to 577 variants. So, okay, well, now we have 577 variants that both samples are being called on. But the next question is, okay, what are the genotype calls for these? And because we're looking for that autosomal dominant inheritance mode, for this case, we want to make sure that we look at the AR call, so the call here on column 9. And in this case, what it does is it says, okay, well, how many samples are being called as a heterozygous change? And so once again, we can click on the activate by threshold, and we can set this equal to 2 because you have 2 samples that you want to be called. And you can see that some of these have 1 calls, 2 calls, where 1 could be for the reference or 1 could be for the homozygous recessive calls. We essentially want both heterozygous changes to be present in both of our samples. And before we hit OK, one of the features here, you can see that they inactivate or activate certain rows or certain columns depending on how you filter and how you analyze. What's important here is that if you don't check this for the secondary stage of activate by threshold, you're actually going to lose all these variants that you already actually inactivated earlier with the call rates. So you want to always make sure that you're only filtering with the active rows, which are the ones that are actually still bolded. So we're going to hit OK. And once we do that, we're down to 314 variants, right? one of the cool things about SVS as well is that, you know, I still have a list of 1,900 variants, but I'm only interested in the samples that actually are not bolded, meaning that they're still activated. So you can actually subset this information out and just kind of create a separate spreadsheet, if you will. And you can click this row subset spreadsheet and actually spit that out for you. And I'm just going to close the window behind it. And so now we're down to 314 variants. But one thing that you can actually see and see the difference is that in the initial master file here, our samples were listed in the rows, and then all your variants are listed in columns. Well, there's no way for us to understand, okay, well, what are the calls for these exactly based on the master spreadsheet? So you actually have to make a secondary spreadsheet that actually talks to each other. And the reason why that's important is because here, all the call rates and information there are going to be on the columns, but all your sample variant calls are on the rows now. And so one tool that SVS actually implemented is called Select. If you go under Select, it's called Activate or Inactivate based on second spreadsheet, because this master file here, once again, has 1,900 variants, whereas we're only interested in these 314 variants, right? So we need to make sure that we can still get all that information out, because your master file here contains all your marker map information, whether it's the gene names, what the filter results were, or even RS numbers that may have still been lingering. So we do activate or inactivates based on second spreadsheet. And this part, I think some people caught on as faster than others, but it's actually kind of simple once you get the hang of it. But essentially you say, this spreadsheet here, the master spreadsheet, I want to make sure that the columns, which have the variants, the columns to be called active, so you want to make sure that they're active, based on the active spreadsheets of the bottom one down here, but you're basically basing it on the rows, because the one thing that it can talk to each other with is the variant location here. So one, this position, and then also the variant position as well. And so you want to select the spreadsheet that we just created, which is here, called marker statistics row subset. And so once again, we're going to set the state of the columns up here based to active based on the information of the rows of the second spreadsheet. And so we hit OK. And you can see now a lot of these variants are now inactivated, and we're down to 2 samples times 314 variants. So once again, we're going to actually subset the columns, because we want to take away all the things that are inactive and just keep the things that are active. And so now you have a condensed list of 2 samples with 314. And let me just switch back to the presentation. And so now we use marker statistics, and kind of allows us to set the criteria to say what samples should have what genotypes and at what call rates, right? And so we got it down to 314 variants. So the other tool that we thought was useful, and I think different groups can also use it as well, it's called Filter by Gene List. And what Filter by Gene List does is you create a text-delimited text file, tab-delimited text file, and you can actually create a list of candidate genes, whether it's cancer panels, or whether that's retinal dystrophies, or whether that's, in this case, eye genes. And you can actually filter with the variants that you currently have in your list based on the gene names. And so what I did was actually imported from National Eye Institute. They had a list of genes that are associated with any ocular diseases out there listed to date. And so what we're actually going to do is we're actually going to walk through and use that list to help us filter that 314 down to a couple more. So here we have 314 variants, and so one thing that I've already done actually is imported that text file that contains my eye disease gene list. And again, it could be for cancer panels, it can be for whatever study that you're working on, but it could actually be used for, once again, you know, the utility here is that if you already have a candidate list of genes that you're actually interested in, or working on a panel, for example, this would be very useful. So I literally downloaded all the information that's available from the NEI, and what we're going to actually use to crisscross is actually the gene symbol here with the master file that we had here, which under marker maps contains the gene names. So essentially, Filter by Gene List will actually sit here and go, okay, out of the 314 variants, which one of these falls within a gene that's implicated or known to be associated with eye diseases? And this can be simply done with a magical click of a button. Under select, there is filter by gene list. And just giving you guys a heads up as well before I forget, one of the things you can probably notice is that some of these things down here are italicized, whereas these are just normal. These italicized scripts here or functions are actually add-on scripts that Golden Helix will help create for you, or they created over time with other users. And so what's great about that is you can always add more scripts to what you do. And again, for those that have no experience in scripting or creating Python scripting, I think these are great tools. And I'll talk about a little bit about the customer service that we've kind of experienced later on. But filter by gene list here is what we're going to use. And so it's saying, okay, well, this master file, it needs to figure out what spreadsheet you're going to pull this information from. And because I already have a list of genes here, we're going to use that. And it's going to say select the column. So you can see there's all these columns here that gives you mRNA accession numbers, Entrez ID, Unigene, et cetera, et cetera. But we only want it to look at the gene symbol here. And so once you do that, marker track, select track, it's going to be on GRCH37 Homo sapiens, and you hit OK. And voila, it says 58 markers were found in the specific gene list, and 256 were inactivated. And once again, this isn't like a end-all be-all tool that you have to use, but if you're down to a couple hundred and you want to get it down even further, this is just a great tool to use. And so again, it unchecked a lot of these variants, so we're going to subset the 58 that's remaining, that's still in the running for the candidates. And let me just go ahead and close these out. And so, yeah, we're left with the variants that have passed through using the gene lists that we have with eye diseases. And so we dropped it down from 314 variants, and now we've got it down to 58. So I think the next cool tool, and I think I encourage everybody to use this, is that I think there was a recent release not too long ago on the functional prediction website, DBNSFP, I believe, 2 weeks ago. And so Golden Helix was nice enough to download and upload that data for us to use. What's great about filtering by non-synonymous functional predictions is actually uses in silico software tools like SIFT, Polyphen, Mutation Taster, Mutation Assessor, Fathom, to predict the change that's occurring with the amino acid change that you're seeing with the mutation. But then it also contains JERP++ and PhyloP, which actually looks at conservation scores. So what's great about this is if you're down to 58 variants and your PI is like, I want it down further, or you think that you can get it down further to maybe perhaps looking at genes that are possibly damaging or disease-causing, this would be a great tool for you to use as a filtering criteria. Now, one thing that I think if for those that are experienced using some of these prediction sites, you can go to each website and actually input your data or FASTA sequence and pull that information out. But the great thing about this prediction database here that Golden Helix uploads on the software for you is it contains all 7 of them simultaneously. So whatever mutations or list of variants that you still have left, it'll crisscross against all 7 prediction tools if you wanted to do so. And as some of you guys have experienced with this, each prediction tool actually tells you something different, right? They have different algorithms that calculates the likelihood of a mutation or an amino acid change to be damaging to the protein. And so this will be a fine example where row 1 actually shows under SIFT prediction tolerated, but then Polyphen 2 and Mutation Taster says that it's probably damaging and disease causing. And so I think we have to be sensitive to that kind of material and kind of use this as a cautionary method to filter. But again, it's also a useful tool as well. And so to go along with that, what we're going to actually use to do so is actually use a tool called filter by multiple columns. And what filter by multiple columns does is, for example, when you have 7 different prediction tools, you can state the fact that, hey, at minimum, I need X number of these categories to be met at a certain criteria that I actually set it at. And what I mean by that is actually you can say, hey, I want at least 5 of the 7 prediction tools to tell me that it's damaging, or you can say 1 out of 7, or you can say all of them have to be. And for the examples of this webcast, I'm actually going to show you that, although these prediction tools actually use different predictors, what if we looked at the fact that perhaps I can set a criteria that all 7 of the prediction tools have to meet the criteria for it to pass to the next stage. And so I'll walk you through that. And so once again, we're back to 58 variants here. And so again, it's currently under an add-on script that I have, but I think with a new release, it'll probably be in the regular columns here. So when you open filter by NSFP tracks, it gives you a couple of information. So it tells you perhaps you can actually annotate variant, you can annotate variants, you can annotate and filter variants, and you can even ask it to remove the non-annotated variants as well. In our case, we want to have some control as to how we're going to filter or how we're going to use some of this database to help us filter. So we're actually only going to ask it to annotate the 58 variants with SIFT, polyphen, mutation taster, et cetera, et cetera, et cetera. And so we're just going to hit OK on this after we click on annotate variants. It's going to think for a split second, and it's been able to annotate all 58 variants. And so once again, you can actually see that the markers actually become rows now, and then it gives you information that I think a lot of other tools probably don't, and it gives you alleles present, what the reference alternate alleles are. It gives you the strands, the codon changes, and once again, we come down to column 9 through, I think it goes all the way to 20. And so we can actually use some of these predictors and filter based on what we want. And in this case, I'm going to say, I'm going to give it the most stringent criteria, and that means essentially that we want to make sure that all 7, it has to pass through all 7 of the prediction tools for it to go to the next stage. And we're going to use filter by multiple columns. And we're going to add columns here, and you can choose from a vast array of columns. And in this case, we're using it for the database here, but you can use it for any other Excel sheets or kind of these pop-ups that you get when you're analyzing using SVS. So we're going to check off the predictions. So for polyphen, SIFT, mutation taster, mutation assessor, Fathom, and JERP and PhyloP. And we're going to hit OK. And 1, 2, 3, 4, 5, 6, 7 tools there. And then we're going to hit next. Now this is the part that I think, you know, different groups or different, the way you want to get the filter down to the nitty gritty all depends on your study design. But up here, the first drop down box actually says activate rows that passed what? At least 1, at least 2, at least 3, all the way up to 6, or you can do all. And once again, you can set that criteria based on any of the prediction tools here that you want. So in this case, once again, we're going to do all of the tools have to meet that it's damaging or a question mark. And the question mark actually refers to some of these tools actually can't predict premature stop codons, or it just has some algorithm that it just cannot predict. And so the question mark actually sometimes has a value that's non-integer. So it could be a negative, or it's a non-positive number, so it could be a negative or something like that. I think you can contact Golden Helix and they can tell you the exact algorithms. But essentially it could include nonsense mutations, so we're actually going to keep those. And we're going to check off for polyphen, possibly damaging, probably damaging, and leave benign alone. Mutation taster, probably do the same thing here. Disease causing automatic. Mutation assessor, we want to make sure that it's predicted to be functional, highly likely, and also medium. I'm going to stay away from low and neutral for now. And then Fathom, we're going to do question mark and damaging. And so keep in mind, JERP and PhyloP are conservation tools and scores. And so I think from what I've seen, that anything greater than 0 or a positive value means that it's somewhat conserved across species. So we're only going to be interested in things that have a greater than 0, so anything that's a positive number in terms of conservation. And once you're comfortable with all the things that you checked off and kind of make that assumption that all 7 tools have to meet these exact criteria to pass, the 58 variants that we started off with, if you hit OK, it's actually now going to dwindle down to 9. And so we go back to the slide. And you can, again, once again, subset this out and then crisscross it with your master file to make sure that you have a master spreadsheet that contains all your information. And so we filtered at the very bottom there, 58 variants, got it down to 9 variants, and we used the filter by functional predictions and also used filter by multiple columns to help us use some of these public databases that are out there to help us predict what's going on with some of these changes. And so we got it down to 9. But once again, we come back to the whole theory of, well, were we able to actually find that needle in the haystack? And in fact, we were. So we actually screened all 9 of the variants, or actually we screened more just because this new data set actually reduced it down. I think we originally started with 50 variants, no matter what databases that we were using. But we found a nonsense mutation on exon 2 of COL2A1. And it's actually co-segregating with disease. We screened it against a bunch of controls and looked at other public databases, and it's not nowhere to be seen. And it's actually interesting because exon 2 mutations is actually predominantly ocular-only phenotype in Stickler Syndrome patients. And obviously, I can't disclose the exact mutation just because it's currently in submission. But here we can actually say that, hey, you know, we were able to use SVS to kind of help us find that needle in the haystack. And so what are some of the challenges and opportunities, right? I think when you're filtering, I think it really depends on your phenotype. And the reason why I say that is because there's a lot of tools that we actually didn't really use today, where I think Dr. Kaufman or perhaps Gabe in his talk actually talked about. But we didn't use known DB-SNP databases. We didn't use NHLBI's 6,500 exomes for controls. We didn't use 1,000 genomes. Obviously, we didn't have a necessity to use compound heterozygous analysis, but that feature is actually in SVS, which really helps for recessive models. Runs of homozygosity can be analyzed. You can use principal component analysis and do association tests also on this software, and which we haven't really gotten around to, but we really are interested in doing so. And one thing that we actually didn't use, and probably some of you guys have caught on, is that we actually didn't use any of the QC metrics when we filtered using, for example, quality depth or anything like that, because what we realized was that just based on this information here, we got it down to 9 variants. And so, you know, what I think is for the second point here, it's not really what's right or wrong, but I think what we have to focus on is what is the most efficient way, right? And so if we're filtering by functional predictions, actually this is supposed to be 7 here, but if we're supposed to be predicting by functional predictions all the way at the top, well, these are prediction tools, so you're not 100% guaranteed that they're going to be truly accurate unless you do molecular work. So you want to place things that are up top, that things that you can control. So if you know that you're looking for a rare variant that's a rare coding variant, then you want to make sure you filter with that. You can use different databases aside from common DB-SNP 137. You can use 132 if you're a little bit more worried about the private mutations that are involved in some of these newer databases, or you can use other tools as well. And then variant classification obviously stays at the top because you're looking for something that's coding, et cetera, et cetera, et cetera. And it kind of points to the third point here, which kind of points to the fact that we want to make sure we minimize some of the reanalysis, right? So again, if we kick in like filter by gene list all the way at the top, what if the list of variants we got it down to 20 was not it? Then you actually aren't able to kind of take a step back and go, what's my secondary approach? What's my tertiary approach? And I think with more and more exome sequencing being done, and then, you know, depending on your staff size and depending on your resources, you may not have the power or the capability to screen 500 mutations off the bat. And so you have to come up with ways to actually get it down to a manageable size. But that manageable size obviously dependent on each group and each study. But once you filter, let's say, for example, we got it down to 9, and if our variant wasn't in the 9, well, we have a very secondary approach to say, okay, well, then we can look through the 58 variants and then kind of analyze those or validate those via Sanger sequencing. And so you want to kind of think about how you want to set your filtering criteria to minimize those types of reanalysis. And I think also understanding your study design is obviously truly important for what you do. So just want to briefly mention some of the points of pros and cons of what we've experienced with using Golden Helix. And I'll start with some of the pros. Customer service, I think, is great. I just want to give a special shout out to Greta, because I think she's been working very closely with our group to kind of help us write scripts and work with that. So one of the great things I think, and I don't see this with a lot of companies, is that, you know, for example, if I have a question, I go, I want to filter it by this way, but there's no tool that you guys have thus far, or there's no script that can do that. We can actually contact them and explain them the situation, and they can actually do that. And I think that's where the add-on scripts keep piling on if you go to their website, is because people come up with these new ideas, and it's ever so changing. And I think, you know, the company actually works with you to kind of come up with these scripts, and hopefully other groups can actually use it to apply for their studies as well. I think there's opportunities for everyone to participate, and I think the reason I say that is because we have, and I'm sure a lot of you guys as well, have students and volunteers that kind of come through and walk through your lab. And if you have a software like this and you give them a certain project, for example, a trio or 5 cases or whatever it may be, it gives, I think, these individuals, especially the younger generations, to kind of work on their work on their skills in terms of, okay, well, this is the criteria that I have. I know it's autosomal recessive, and I need to filter it down by this tool, these tools, and actually allows them to use their brains to kind of help you filter. And I think sometimes these guys are the ones that have the greatest expansion of minds, and they're naive to a lot of these research studies that some of us have been in for years and years and years. And so a lot of times, sometimes these scripts that we kind of think of comes from those individuals themselves. And documentation and internal QCs are really important. And I don't know if you saw it on the, let me just go back to the SVS, right here on the right side, it actually keeps track of what user ran what different type of script for whatever Excel sheet, or in this case, these little nodes here. And so what's important about this is then it allows you to have QC controls and documentation of exactly what was ran. And so if there's a new database coming out, so for example, if last year you were screening, filtering by dbSNP132, and now you want to do dbSNP137, you have the tools to actually look at it and go, okay, well, somebody else did this and this on this project. It actually allows you to kind of share that kind of information. And as I kind of mentioned with scripts, I think there's endless possibilities. I think it's obviously the way we filter, or the way some of these public databases keep changing. I think Golden Helix is really up to date onto some of these public databases, and it's always going to be changing, right? And so cons, obviously the learning curve is huge. I think it took a few weeks for each individual to kind of go through, but once you kind of go through the nuances and understand the terminologies of some of these tutorials and also some of these filtering criterias, it's not too bad, but there is obviously a learning curve. And I think a lot of smaller labs and smaller organizations have issues with cost, right? And some of these softwares are not necessarily cheap. But I think one thing that I do encourage and kind of talk about is you should probably, if possible, look for other collaborators or look for other individuals who are working on similar projects. And perhaps you guys can all chip in together and purchase a concurrent license. So here at the Center for Human Genetics, we currently have 2 concurrent licenses. And what's great about that is because we also have a team in Singapore, because they're 13-hour difference, while they're using it at night and while they're using it during the day and we're asleep, it's vice versa. So during the day, like for example today, they're not using it at night, but we're actually using it. So we actually, for our group, we actually maximize almost a 24-hour usage of the SVS softwares. And I kind of put continual updates as a con, and the reason I say that is obviously we started off without using some of these dbSNP137 and using the newest non-synonymous functional prediction tools. We had it at 50 variants, but obviously with new updates that's always releasing, you may have to redo some of the steps or redo some of the analysis. And I think continual updates can hurt you, but at the same time, I think it also has its strong benefits as well. And so there's an infamous diagram that states that the theory in project management, actually, within an organization, and that there's often 3 constraints that affect quality, which can't be changed without affecting each other. And oftentimes people struggle with the notion that you can only choose 2 of the following. So either fast and good, good and cheap, or fast and cheap, right? But if we kind of take a step back and take a look at what's going on in sequencing and in the field of sequencing, then I would actually beg the question, and actually I think sequencing actually defies some of these theories, right? So if you take a holistic approach of what's transpired in the field thus far, in the past couple of years. It's amazing. Like, we've gone from screening one gene at a time to screening your entire genome and having it analyzed in a few weeks, which I think it's considered fast. I think service providers and academic institutions are creating best practices for some of the bioinformatic pipelines, for example, GAC-K for variant calling. And with different applications of NGS, which I think can work on the good portion there on the right. And lastly, and as many of you are aware, the cost of sequencing has dropped at an exponential rate over the past several years alone. And so I think there's the endless possibilities to refine these odds and provide a sense of hope and purpose of what we're doing is very, very important in the field of sequencing. And so lastly, so will we ever see the light at the end of the tunnel? And personally, I like to consider myself on the optimistic side and say that there are lots of work to do, but I think it's very, very hopeful. The strengths in short turnaround times, low cost, and relatively easy data interpretation places us in a position to do more and more with this information. The challenge is to reduce the gap between, I think, genomic interpretation and also medical interpretation is certainly vital in taking this to the next level. And what I mean by the next level is, as healthcare shifts, I think, currently it's shifting towards population-based health management and evidence-based medicine. How can we actually leverage some of this genomic information to help patients and reduce some of the burdens surrounding healthcare? I think it's exciting times right now. Our lab is actually currently sifting through 84 exomes at the moment with different phenotypes, and we're definitely excited about some of our findings thus far. We've taken several phenotypes to the animal modeling stage and have seen some encouraging results from our findings and hope to share some of this upcoming publications. And if you're actually in Seattle from May 4th to the 9th, there's an iConference called ARVO, and so we can actually certainly discuss with you in person then if you're going to attend there. And lastly, it's not a one-man team, and I certainly like to acknowledge everyone who's helped us kind of walk through and work with us with exome sequencing. And once again, special shout out to Greta, because I think she's been the most helpful in terms of creating some of these scripts or kind of giving us more ideas as to some of the insights as to how we can filter. And so hopefully, with everyone here and their contribution in making gene discovery, I think it's definitely made it a little bit more fun, challenging, and exciting. So with that, thank you for listening, and I'll be happy to answer a few questions.
All right, thanks, Khanh-Nhat. That was a very enlightening presentation. I appreciate your candidness on the feedback for our software, and it's always really good for us to see how our tools are applied in the field, so I really appreciate that. I'll give people a couple seconds here to submit some questions. We already have some. I'll go ahead and start asking some of the questions that came up a little earlier during the presentation. Could you please elaborate the extent or measure of variations expected in exome data, including types of parameters, statistical measures, etc.?
Right. So I think for our case, we had 75, I think we started off with 74, about 73,360 variants. But one of the things that I guess I failed to mention here is actually we took, each sample here was actually analyzed separately on their own. So there was a lot of overlaps in terms of some of the variants didn't have a call on one but had a call on the other. But I think roughly in terms of coding variations, I think you're looking at somewhere around that 25 to 35,000 variants. And I think depending on the population, I think I read somewhere recently where I think with African Americans there may be a little bit more private mutations that are involved. So yeah, definitely be sensitive about that as well.
Okay. Next question here. First a statement. Nice work. Congrats. And the person asks, I'm not sure that I understand how it is you knew you were looking for heterozygous mutations. Since it's autosomal recessive, wouldn't that suggest a homozygous mutation?
Right. So in this case, it's actually autosomal dominant, and that's what we have. I have a pedigree here. If I can sort of see here, is actually we have it's 4 generations, but here as well, I wrote that it's autosomal dominant, and that's why we're looking for a heterozygous change in this case.
Okay.
But absolutely, if it was a recessive model, then we would have indeed looked for homozygous changes, or even compound heterozygous changes.
Yeah, so there's been a couple comments, which are some of them in forms of a question, but here's one that's kind of summarizes some people's thoughts here. I understand the need to narrow down the variants, but the gene list filter has a drawback. You can only discover what is already known. That was a comment made by somebody else earlier in the presentation as well about your particular filtering strategy. And really the question is, are you limiting yourself by the filtering strategy that you decided to go after?
No, I mean, absolutely. This is part of the webcast here is I wanted to showcase the tools that you can actually use. So in fact, actually, if I do 314 variants here and I actually exclude the number of gene lists, we're still down to a manageable size. I think it floats in the 40s or 50s in terms of just based on functional predictions. But this tool, I think, is more or less useful for individuals that are looking for the known genes. But if you're not actually looking for it, absolutely, I would take that out. And you could obviously limit yourself, but I think one of the constraints that we have in terms of research side is if a PI wants it down in the teens or 20s, then we have to come up with tools or come up with ways to kind of have a staged approach in some of these screenings. I mean, I can't. There's no way for me to say I have 314 variants. How do you want to screen that? Do you want to do Sanger sequencing? Because by that time, then perhaps we could have included another sample for exome sequencing and kind of figure out what the cost benefit of maybe exome sequencing another sample would have helped. Whereas spending more money on 314 variants to use for Sanger sequencing validation. But I absolutely do agree that the gene list may limit you on what you do, but at the same time, it could perhaps give you a quick, stringent way to kind of look at, hey, is there novel mutations or perhaps novel genes or not, I'm sorry, not novel genes, but novel mutations that isn't already implicated with a certain phenotype or certain group of phenotypes. So again, it's just a tool there, but I do agree with some of the comments there.
Yeah, and I would also just say that this field is progressing rapidly. As you mentioned, Khanh-Nhat, one of the reasons why there's so many changes to the software, so many updates, is because there's new annotations coming out all the time. People are coming out with new ways to filter. And I would just encourage you on our website where we do have these webcast recording, there's a number of webcasts by Ken Kaufman from Cincinnati Children's and Gabe Rudy from Golden Helix here, just discussing different approaches or different ways that you can filter your data. So what's really nice about our software is it's very transparent and flexible, so you can try different things to cull that list down in a way that makes sense for your research goals. All right, let me look back through the list of questions here. Somebody asks, and this I guess applies to the number of different studies that you've done. How many times have such approaches not been successful in your experience? Maybe you can elaborate on your own experience as well as maybe other ways that your colleagues have done this, Khanh-Nhat.
Right. I think one thing that I think we have to be sensitive to with exome sequencing or whole genome sequencing or any other of these new technological advances is that there's a likelihood that the needle in the haystack is missed, right? And so a lot of times, when we're working with recessive models, there are trios that we actually work with or cases that we work with that are inherited in a recessive model, but we actually don't find anything. And we get it down to those 20 variants and we don't see anything that co-segregates or that's biologically relevant. And so we actually have to keep taking a step back and kind of reevaluating perhaps some of the filtering tools. So if you're working with something that's, for example, in high-grade myopia where 3% of the population actually is high myopia with a refractive error -6 or greater, then you actually have to be sensitive about that, and perhaps that in the general population, some of these variants that perhaps have RS numbers with less than 1% allele frequencies could be still candidates for co-segregating in your families. So you have to be definitely sensitive about that. But I mean, for us, we've been fortunate because we work with so many different types of families and also so many cases that there's so many projects to go on simultaneously. So if we hit a dead end, we actually move over to another one and kind of work our way that way. But definitely, we have had some roadblocks along the way for sure.
Okay. One person asks here a clarifying question that I believe you covered in the presentation, but does the mutation you found, the particular mutation you referenced earlier, does that explain the variability in the phenotypes between the family members?
No, it doesn't, and that's a really great point. However, one thing that once we found that novel mutation, the interesting thing was on specifically exon 2 is ocular only, supposedly ocular only, but there were a couple papers who they don't mention it expressly in their papers, but they actually did find individuals, for example, with cleft palate or musculoskeletal diseases as well, in addition to the ocular. So I think there still needs to be some more studies that needs to be done, but I think the variability is also documented well in other literature.
Another question that I can answer here, somebody asks, can Golden Helix SVS annotate whole genome sequencing data? If yes, are there limitations of how many sets of genomes can be processed? I would answer that with yes, we can very much annotate whole genome sequencing data. The extent that we can annotate is based off of what's available in the field today, but we do provide a number of tools. Khanh-Nhat, I don't know, do you have any whole genome sequencing data yourself that you've used with the software?
We're actually working with a collaborator, and he just gave us some whole genome data, but we haven't actually played with it. One problem, I actually try to upload that data, but one thing that I don't know if other groups use what computers, but we actually use Windows 32-bit, and we have seen that using a Windows 32-bit slows down sometimes analysis or locks up the software if you have a large data set. So for example, in whole genome, I know it's locked up a couple times, so I actually have to move over to a 64-bit computer. So I would actually be careful on that if you guys are whatever computers you're using, but we're in the midst of actually analyzing that as we speak.
Yeah, and I would say that the whole genome sequencing is significantly larger in terms of the size of the data sets, and so a larger computer or more memory is needed to analyze those in a reasonable fashion, yes. But thus far, we haven't really come across any hard or concrete limits in terms of the number of samples that we can analyze with the software. It really depends on the hardware that you have. All right, looks like we got time for a couple more questions maybe. There's actually quite a few, so let me look through these really quick. Somebody asks, and this is kind of an open question, but how can we use whole exome sequencing for looking for genetic modifiers of complex diseases?
Yeah, that would be a very open-ended question. I'm not too familiar with that since we work with a lot of Mendelian.
Yeah, and I would say that Gabe Rudy, actually, our VP of Product Development here, he's been analyzing some of his own personal exomes for himself and his family. He's got a really good blog post on our website. If you haven't seen it yet, it's on Our Two Snips, where he actually talks about some of the issues with exome sequencing in complex disease. A very fascinating blog post, and there's also a video to accompany it as well. All right, let me look here. One last question. So you got 9 candidate variants already, but what is the benefit to using Genome Browse for these data? That's a good question.
Yeah, that's a good question. Here we didn't use it just simply because we got it down to 9 variants, but I think the utility of it, even with 9 variants or even with 50, and we did actually use it, I just didn't cover it in the webcast, but the Genome Browse will actually show you the actual coverages between your reads for that mutation that you have. So for example, if you take a look at that mutation that we're actually interested in, the coverage was about 25x coverage depth, and it was split between one allele 50% and the other allele 50%. So you can actually physically take a look at it. And sometimes there are issues where there could be some bias towards one allele than the other. And so those are kind of more visually confirming, the ways to visually confirm some of these mutations. And you can use quality metrics to help you filter that out to begin with. But I think visually looking at it also helps as well. Simply because out of the, for example, in theory, if you get it down to 9 variants, perhaps 3 of them are at 5x coverage. And if it's at 5x coverage and you're actually looking at it visually, does it actually make sense to expend your money in validating those? But I mean, I do have proof of concept that, at 4x, we were able to discover a nonsense mutation for another phenotype. So I think it goes both ways there.
Yes, well, thank you, Khanh-Nhat, again. We're kind of at the top of the hour here, so we'll go ahead and turn the presentation off here in a second. I would say that there are many more questions. We didn't get to all of them by any means. That's always a good sign. We always do our best to either answer some of the questions ourselves after the webcast is over, or we'll pass them off to Khanh-Nhat as well if they're geared specifically toward him. And thanks again, Khanh-Nhat. I appreciate it.
Thank you.